Hello. Hi, guys. Welcome back. Today we are going to be talking about DNA mutations. Not all mutations are bad. So that's the moral of the story. All right, so I am going to share my screen and we'll talk about DNA mutations. All right. Oh my, interesting. Let's try this instead. All right. Okay, so this is the last part of the PowerPoint that's about transcription and translation, chapter 17. And we are now talking about DNA mutations that occur in DNA. So there's three main types of mutations. Well, actually, let me rephrase. The first kind of mutation I want to talk about is called a point mutation. All right, a point mutation means you're changing a single base pair. So this is a single nucleotide change or a single base pair change, right? So you're changing one of those bases in DNA, A, T, C, or G, to something else. And so there are three kinds of point mutations. The first is called a silent mutation, where you have no effect on the protein at all, and that's why it's silent. The second one is called a nonsense mutation, and this leads to a premature stop codon. So it's a mutation that changes a nucleotide that creates a stop codon, right? And then the third one is called a missense mutation, and this leads to a change in an amino acid, and therefore it can lead to a faulty protein, all right? So if we look at these, let's start here with our normal non-mutated DNA. So our DNA is ATG here. Its corresponding mRNA that would be transcribed would be complementary base pairs. So that would be UAC, and that would create a tyrosine amino acid, and that would lead to our normal protein. A silent mutation, number one here, we are changing this A, it has been mutated from a G to an A. So we had a G to an A single point mutation. Hold on, sorry. Gotta turn off my tea so it doesn't ring in your faces. All right, so here is a single point mutation. We had a G to an A. But what you'll notice is that the codon is different in the mRNA. So there was a mutation. However, that codon still encodes for a tyrosine amino acid. So there's no change, right, in the outcome of the protein. So that's a silent mutation. Yes, there was a change in the DNA. No, there was not a change in the protein. That's a silent mutation. The second one is called a nonsense mutation. Here we had a G to a C, and what you'll notice is that that changed the codon, just like it changed in number one. However, what that codon was changed to was a stop codon, all right? So remember, and again, you would have to look at that codon chart to see the stop codons, UAG, UAA, or what's the other stop? Or UGG. So those are the three stop codons. So if you have one of those codons, that will cause a stop when you get to the ribosome. All right, so you would have an incomplete or truncated protein, right? The third kind of mutation, in this case, we saw an A went to a G, and you see that the first nucleotide in that codon has switched. Therefore, you get a completely different codon, which encodes for a completely different amino acid. All right. So here we went from a tyrosine to a histidine. That causes a mutation in the protein, which often leads to problems in folding or problems with the protein function. So that usually leads to a faulty protein. All right. Another kind of, so those are the three main point mutations. However, you can also get a single a single change in the DNA by a single nucleotide insertion or a deletion, right? And that will lead to what's called a frame shift mutation, right? So a frame shift mutation is caused by an insertion or a deletion of any number of nucleotides that is not divisible by 3, right? And why is that 3 important? Because that's what the codons are, base pairs of 3, right? So if you have a insertion of a single base pair, that is way worse than an insertion of 3 base pairs. 3 base pairs would just leave, have 1 change in the amino acid, whereas if you have a single base pair mutation, 1 base pair, that will cause every single amino acid downstream of that to be wrong. All right, so let's look at that. So if we have our normal translation here, if you add a single base pair insertion, so let's look at where we added here. So here we have ACG, AGG. Here we have ACG, AGG. Where did we add here? Let's see. So if you added, so ACU, ACU, GCA, GCA, UAC, UAC, C. Okay, so we're adding, it's the exact same sequence, but what we've done is that we have changed the frame shift, all right? So what you can see is adding a single amino acid will cause the whole thing, every amino acid downstream of that to be wrong, right? So instead of starting our codon here with ACG, we've kind of shifted it, and now we're having the frame, the codon start with a completely different one, all right? So those are, every amino acid downstream is bad. The same thing happens is if you delete an amino acid, all right? So here we added an extra one. Here we are deleting an amino acid, and now every amino acid downstream is wrong, right? Those are frame shift mutations. So I want to give you an example in terms of a real life disease. So here's a mutation that causes the disease called sickle cell anemia, that causes the red blood cells in your body to be kind of sickle shaped, as shown here in this image. Normally you have these nice kind of disc shaped red blood cells. With sickle cell anemia, you have these long kind of shapes. And what that actually causes is that normal hemoglobin, the protein within our red blood cells, is normally kind of shaped like this. Whereas in sickle cell anemia, these hemoglobin clump together, and that's what causes these sickled red blood cells, and it causes a huge number of problems. Side note, there's now really cool, very promising data shown that CRISPR, which is DNA editing, can really help these individuals. But that's a side note. All right, so let's look at the DNA sequence. So here's the DNA sequence. Here it says this is the start of the coding sequence. So that's the part that's going to make your proteins. All right, and here's the DNA sequence. And if you look at the resulting amino acid sequence, you see that normally we have a glutamic acid in this position, right? If we look at the DNA sequence of the mutation, which is down here, we'll see that normally there's a T there. Here, it has been mutated to an A, right? So that base pair has switched, right? Still AT, they still pair up well, but the coding sequence is fundamentally different. And therefore you get a valine in that amino acid spot. And only one amino acid change causes these really huge physiological effects, right? So here is a good thing that you guys should try. So after consulting genome databases, you find that a human hexokinase amino acid sequence begins with methionine, trip, lys, trip, met. But the protein you are working with is this. And you can see that there is a difference there, right? So you can see that there's a lysine versus a methionine here. There's some sort of change in the protein sequence. So your job will be to write the DNA sequence of the non-template strand for the beginning of the actual non-mutated hexokinase. All right, so you are going to have to figure out what the DNA sequence was based on that. All right, don't forget to include your 5 to 3' ends. There is an answer here online. All right, this is a good thing to try to recognize DNA mutations. So for example, what is the mutation here? That is a frame shift, right? If you look here, here's the original sequence. What happened? What kind of mutation was this, right? So if you'll see, we have an A to a C. So that was an A to a C, and it caused a change in the amino acid. So that would be a missense mutation. Here we had, and this one down here, we can see that there was a single, what happened here? So it looks like this was an addition, right? So we have CAG, CCC, CAG, an additional T, CCA. So this was an insertion, a single amino acid insertion. Here we had, again, it looks like a single amino acid change, but there was no change in the accompanying protein. So that would be a silent mutation, right? And here it looks like we had a premature stop being in. So that would be what was that called again? Missense, silent, frame shift. What is it called? Nonsense. Sorry. Stop is a nonsense mutation. All right. So just to give you a sense that those are the kinds of things that you'd be responsible for knowing. All right. That's DNA mutations. Remember, at the end of the PowerPoint, as always, lots of practice questions. All right. Toodles poodles. Sayonara. Ta-ta for now. Bye.